We'll start the next portion of the course looking at macroevolution, that is, the evolutionary processes that create new species from their ancestors, and more specifically, how we recognize which species are related to which. This chapter also looks briefly at humans' mammal relatives. Chapters 6 and 7 will be devoted to primates. So to begin with, if we're studying macroevolution, and macroevolution is the creation of new species, it should be obvious that we really need to understand the species concept and how species can be identified and described. The first thing we need to cover then is biological classification. Classification, of course, is just the process of putting things into classes. Taxonomy is the branch of science concerned with the rules of classifying organisms on the basis of evolutionary relationships. To put it another way, taxonomy is the study of how to do classification of living things in the best, most useful way. We can't tell if new species have appeared unless we can list out the species both before and after and see that those lists are different. To do that, we need to be able to classify individual animals into species. Traditionally, those categories have been based on the physical similarities between different animals. Linnaeus was the first person to really systematize the process, and he introduced the idea that categories should be hierarchically nested. Small categories contain a few species that are very similar to one another, and those are then lumped together into larger categories that contain more species, but with more differences, one from the other. Linnaeus based the larger and smaller categories of his system on homologies, similarities between organisms based on descent from a common ancestor. This image is a classic way of describing homology. It shows the skeletal structure of several vertebrates' forelimbs. You'll notice that despite the differences in shape and size, the numbers of bones and how they articulate with one another are all very similar. This is because all of the species inherited this structure from their common ancestor, the so-called stem reptile. We specify that homologies exist because of common descent to distinguish them from another kind of similarity, analogies, or similarities between organisms based strictly on common function, with no assumed common evolutionary descent. Natural selection adapts species to their natural environment, so sometimes species in similar environments evolve similar traits, even though their common ancestors lacked those traits. This process is called homoplasy, or convergent evolution. Homoplasy is the process that creates analogy, the result. The difference between homologies and analogies was one that Linnaeus didn't understand because he did not understand evolution. So some of the categories he constructed based on certain analogical traits turned out to not make any sense. For example, he originally classified whales as fish because of the analogy of living in the ocean. Very quickly, however, he realized the problem with that, since whales have more similarities with mammals than with fish. So in the second edition of his book, whales were classed as mammals, where they remain today. This highlights another basic requirement of modern biological taxonomy. It must construct categories of species that reflect evolutionary relationships. Larger categories include all species descended from more distant ancestors, while smaller categories include only those species descended from more recent common ancestors. So let's assume these 4 dots represent 4 living species. These species group together into 2 genera. That taxonomic grouping conveys exactly the same information as saying that species A and B are both descended from species E, while C and D are descended from a different ancestor, species F. Meanwhile, both genera 1 and 2 belong to family 3. That says that both species E and F are descended from a more distant ancestor G. Therefore, with a more properly constructed biological taxonomy, you can easily tell how recently 2 species shared a common ancestor by finding the smallest category to which they both belong. The smaller the category, the more recent the common ancestry. But how do you begin constructing those categories in the first place? How did I know to put A and B in genus 1, but to put C in a different genus? It can't be because I already knew their ancestry, because the main reason to study taxonomy is to learn the ancestry. Today, most evolutionary scientists classify species using the strategy of cladistics. an approach to classification that attempts to make rigorous evolutionary interpretations based solely on analysis of certain types of homologous characters or traits. In cladistics, most homologies are considered unhelpful, and analysis must focus only on certain kinds of homologies. A clade is a group of organisms sharing a common ancestor. The group includes the common ancestor and descendants. Each species defines its own clade, consisting of itself and any species that might descend from it. On this diagram, there are 7 clades, each of the 4 living species, each genus and its ancestor, and the whole family and all 3 ancestor species. Because of this emphasis on ancestors, cladistics is very concerned with differentiating ancestral from derived traits. An ancestral trait is one that was present in the common ancestor of the clade. It is thus a homology between the ancestor and the descendant, as well as between any 2 descendant species that preserve the ancestral trait. Species that share ancestral traits are members of the same clade, since they both inherit the trait from their common ancestor. But other descendants of the same ancestor may have lost that specific trait, such as snakes losing the legs of their reptile ancestors. Snakes are still reptiles, but we can't identify them as such just by looking for the ancestral trait of having 4 legs. What we need to identify is not the ancestral trait, but the derived or modified trait, characteristics that have arisen by changing the ancestral form. Derived traits indicate that the ancestral trait was once present. Snakes have no legs, but they do have vestigial pelvises that indicate legs were once present in their ancestors. Species with shared derived characteristics belong to the same clade, because their traits all developed from the same ancestral form. The more shared derived traits 2 species have in common, the more recently they shared a common ancestor. For example, having multiple different kinds of teeth, a trait called heterodont dentition, is an ancestral trait of mammals. Saying that humans have 4 kinds of teeth, incisors, canines, premolars, and molars, doesn't tell us much of anything about our evolutionary relationships with dogs or baboons. Since they both also have heterodont dentition, we can say that we're related at some level, but not really which. But let's look for a shared derived trait, how many of each kind of tooth. We see that we have 2 incisors, 1 canine, 2 premolars, and 3 molars in each half of each jaw. This is called the dental formula for the species. Humans and baboons both have 2, 1, 2, 3. In their upper jaws, dogs have 3, 1, 4, 2. So humans and baboons share the derived trait as well as the ancestral trait. Dogs share the ancestral trait, but not the derived trait. So we're more closely related to baboons than we are to dogs. In this case, heterodont dentition, the ancestral trait, wasn't particularly helpful. The dental formula is the derived trait, and it was helpful. Cladistics focuses exclusively on derived traits to determine evolutionary relatedness. Of course, the end result of cladistic analysis is a taxonomy, a series of hierarchically arranged categories that consistently reflect evolutionary relationships. Strictly speaking, the statistical processes used to create those categories produce a cladogram, a chart showing evolutionary relationships based solely on interpretation of shared derived traits. It contains no time component and does not imply ancestor-descendant relationships. On a cladogram, at least this style, each horizontal line represents a clade. The vertical lines that branch off the same horizontal line all represent groups that belong to that clade. The lower the horizontal line is on the chart, the more vertical lines branch off of it, so the bigger is the clade. Strictly speaking, cladograms do not indicate which species are ancestral to which others. They just show how closely related species are to one another. For example, this cladogram of the land-dwelling vertebrates shows a close relationship between birds and theropod dinosaurs, the two-footed ones like T-Rex. They belong to the same small clade at the upper right. The reason those two groups are so closely related is that the theropods are the ancestors of the birds, like grandparents and grandchildren. Meanwhile, the theropod bird clade and the other dinosaurs both belong to the same larger clade one step farther down the cladogram. But in this case, the theropod bird group is more like a cousin to the other dinosaur group. The cladogram itself doesn't give us the information necessary to differentiate between the ancestor-descendant relationship and the cousin relationship. All cladistics does is show relatedness. As statistically accurate as that is, it isn't really what our brains want to see when we talk about evolution over time. We want to see a phylogenetic tree, a chart showing evolutionary relationships that contains a time component and implies ancestor-descendant relationships. This phylogenetic tree shows the same groups of living things as the cladogram, but now you can clearly see that the bird branch splits off of the dinosaur branch, indicating that some dinosaurs are ancestors of birds. But the dinosaur branch continues past the branching point, so some dinosaurs are not ancestral to birds. The phylogenetic tree thus contains information not present in the cladogram, and that information comes from the scientist's interpretation of the data. The cladogram is mathematical and exact. The phylogenetic tree is a theory put forward by the scientist to attempt to explain the cladogram's data. Different scientists can look at the same cladogram and construct different phylogenetic trees. Then the next step is to seek more data to test those theories. Okay, so if we want to start studying macroevolution, we need to gather together all the species we know about and then use a cladistic analysis to determine how closely related they are. Then we can take some interpretations about ancestor-descendant relationships and tell which species developed into which others. As I said at the beginning of the lecture, this assumes we have a strong grasp of the species concept. The most popular definition of species today, the one we discussed a few lectures back, is called the biological species concept. It states that a species is a group of individuals capable of fertile interbreeding with one another, but reproductively isolated from other such groups. Speciation, the most basic process in macroevolution, is the process by which a new species evolves from an earlier species. Because speciation usually takes place over millions of years, studying it usually involves studying fossils. Now, fossils tell us what the ancestral species looked like, but there's a problem. We define species according to their reproductive behavior, something we can observe directly for living species. But fossils are just rocks. Rocks don't reproduce. So how can we define a species in the fossil record? We know that when those animals were alive, they must have reproduced, and uniformitarianism tells us that the idea of reproductive isolation was just as applicable in the past as it is today. But if we can't observe reproductive behavior, how can we tell which fossil animals interbred with which others? Well, put simply, and much more directly than your textbook, we can't. Fossil species are not biological species at all, really. They're paleospecies, species defined from fossil evidence, inferred to be reproductively isolated. That is, we've looked at the skeletal evidence and decided that these animals were similar enough to one another, that is, intraspecific variation is low, and different enough from other animals, that is, interspecific variation is high, that they probably constituted a species. We make that decision based on how much variation can be seen in modern similar species, species which are usually considered to be their descendants. This makes paleospecies an approximation of biological species. This concept is as close as we can get to applying the biological species concept to fossils. Paleospecies are theoretical constructs. We can't observe those animals directly, only through the lens of their fossils and their evolutionary descendants. We use these theoretical constructs to help us explain the patterning we see in the fossil record. Paleospecies also present an entirely different problem from living biological species. Because they're theoretical constructs, and because the fossil record is usually so spotty, they're often covering very long spans. Biological species exist at a single point in time, right now. So variation across time isn't really a problem with them. But because microevolution is constant, and because it is the basis of macroevolution, species that cover extremely long spans of time, like paleospecies, may have considerable variation from the oldest to the youngest. Could an early Homo erectus around 1.8 million years ago interbreed with one of the latest Homo erectus about 400,000 years ago? They were biologically similar, but certainly much more different than any two living human beings are today. Now, based on that observation, we might conclude that those two specimens were unable to interbreed, and therefore they should be called by different species names. One is Homo erectus, and the other is Homo whatever. But Homo whatever is descended from Homo erectus. Where do we draw the line to split the Homo erectus species from Homo whatever? If we're going to divide one unbroken lineage into two paleospecies, we'll have to draw the line somewhere, and that will divide a parent of one species from its child in another species. That violates what we know of microevolution, that parents and children always belong to the same species. Paleospecies, therefore, are really not the same thing as biological species. Biological species are observations, data, and paleospecies are interpretations, theoretical constructs, only valid if the theory is valid. Which paleospecies are proper depend on which theory is being tested. Different scholars with different research questions will assign different paleospecies names to the same fossils. The particular labels are only useful as long as they're useful. If you get too caught up in deciding which species label to slap on a particular fossil, you'll likely miss other, more important issues. Keep that in mind as we wade through dozens of hominin species over the next few weeks. Okay, let's look at a couple patterns we see when we study macroevolution. One of the most recognizable is called an adaptive radiation, the relatively rapid expansion and diversification of life forms when they move into new ecological niches. This is when one ancestral species suddenly evolves into many descendants, all with different traits. When you create a phylogenetic tree of this process, you see one trunk split into many branches, with the branches radiating outward in all directions. It's a common pattern in the history of life on Earth. Mammals underwent an adaptive radiation after the extinction of most dinosaur species 65 million years ago, and hominins went through an adaptive radiation about 3 million years ago, which is why there will be so many species names to learn over the next few chapters. But it's important to understand that radiations occur throughout the living world. Understanding adaptive radiation requires understanding generalized and specialized traits. Some animals are generalized. They're adapted to be able to perform a wide variety of tasks and to change their behavior in response to the environment, but they may not be particularly efficient at what they do. Specialized species follow the opposite strategy. They're adapted to do only a few things, but to do them very well. Usually, only generalized species are able to take full advantage of new niches because they are able to change to fit the new environment. Thus, the ancestral species of an adaptive radiation is almost always more generalized, and its descendants are more specialized. Also, there's the question of the rate at which macroevolutionary changes occur. Darwin saw evolutionary change as slow, steady, and incremental. The rate of change did not change. This view is called gradualism and was the common view of most scientists throughout much of the 20th century. It views macroevolution similarly to the tortoise in The Tortoise and the Hare. Once the race starts, the tortoise moves along slow and steady from the start line to the finish line. But since the 1970s, many scholars have come to the conclusion that the pace of evolutionary change is not steady. The rate of change, in fact, changes. There are long periods of stability or equilibrium, broken up or punctuated by relatively short periods of rapid evolutionary change. So this view is called punctuated equilibrium. This views macroevolution like the hare. Once the race starts, he shoots off very quickly, then stops and takes a rest, then shoots off very fast again, then pauses until he reaches the finish line. The different interpretations have some significant effects on how scholars connect species through time. If fossil 2 is a descendant of fossil 1, a gradualist would describe the evolutionary path between them with a straight line. Punctuated equilibrium would describe the path with some kind of curve. More vertical parts represent punctuations, while more horizontal parts show equilibria. Each curve is a theory. The only way to distinguish between these different theories is to find more fossils that fall between 1 and 2 and see which curve they fall along. Macroevolution, of course, usually takes place over the course of extremely long periods of time. Studying these long periods is very hard for human beings. We really lack the mental capacity to understand the time scale on which macroevolution operates. But geologists, who deal with timescales even longer than evolutionary biologists, have systematized their understanding of history into the geological timescale, the division of Earth's history into eras, periods, and epochs. It's this system that evolutionists use to describe large-scale macroevolution. Most important for this class will be the last 4 epochs: the Miocene, Pliocene, Pleistocene, and Holocene. It is since the last bit of the Miocene that hominins have evolved and diverged from other primates. For this unit, I'd like you to know those 4 epochs in that order. Next unit will add numbers to describe how many years in the past each of these epochs occurred. Finally, pay special attention to the sections of this chapter covering mammalian characteristics and evolution. Especially know the 3 major groups of mammals and their differing reproductive strategies. All of this information will help you make sense of the material we cover in the next several chapters as we delve deeper into humanity's evolutionary and biological context. Now, starting next lecture, we'll examine in detail one particular kind of mammal, the primates.